Ladies and gentlemen, thank you for standing by and welcome to the virtual fireside chat with Regeneron conference call. At this time, all participants are on a listen-only mode. After the speaker's presentation, there will be a question-and-answer session. To ask a question during this session, you will need to press the star and the 1 key on your touch-tone telephone. Please be advised that today's conference is being recorded. If you require operator assistance, you may press star and 0. I would like to hand the conference over to your speaker today, Evan Sigerman. Please go ahead.
Thank you, operator, for joining us this morning. It's my pleasure to have with us on the phone Regeneron. And from Regeneron, we have Bob Landry, CFO, Israel Lowy, SVP Translational Science and Oncology, Neil Stahl, EVP of R&D, currently focusing on the COVID-19 program, Justin Holko from the IR team. And just before we get started, I want to read a quick disclosure statement for everyone on the call. By accepting this invitation and dialing into this event, you consent to having your contact information being provided to the corporate presenters so they may better understand their audience and ensure the content of their presentation is appropriate and relevant. If you do not agree with that, please disconnect now and send me an email. And of course, the MiFID II notice, this event is only open to CS clients who are in scope for MiFID II and pay for interactions. And of course, those who are not in scope for MiFID II. It is your responsibility to ensure you are entitled to attend and not receive an inducement. Again, if you do not meet this, please disconnect and let me know. Now that that's out of the way, thank you, everyone from Regeneron for joining us. Are you guys on the phone?
Yes. We're here, Evan. Good morning.
Perfect. Excellent. Good morning. So with that, we have a lot to get through. I want to hand it over to Bob just for a quick introduction and some forward-looking statements. And then I'll speak to Izzy, Neil, and Justin. I'm sure will chime in. But I'm very much looking forward to the conversation with everyone. So, Bob?
Sure. Thanks, Evan. Good morning. Good afternoon, everybody. So our statements today, we will be making forward-looking statements that will involve risks and uncertainties relating to future events and the future performance of Regeneron, and actual events or results may differ materially from these forward-looking statements. As always, I encourage everyone to go to our first quarter 10Q or our year-end 10K to find the risks and uncertainties that are outlined within those documents. Back to you, Evan.
Perfect. That was a quick introduction. So I want to start off with Izzy, who essentially is the SVP of Translational Science and Oncology, responsible for Libtayo, but also all the novel bispecifics and co-stimulatory assets that have been introduced recently. So I want you to kind of frame the oncology program. Tell us where you are and kind of what the overall strategy is.
Sure. Thank you very much, Evan. And good morning to everyone who's listening, and thank you for getting up early to listen. So I'm pleased to say that I've been now at Regeneron for just a little over 10 years. And after years of sort of sowing the crops, we're really excited about now starting to reap our harvest. Before Regeneron, I was at a company called Medarex, which pioneered some of the immuno-oncology revolution, where I led studies in anti-CTLA-4 and anti-PD-1, which was then acquired by BMS. And then I came to join George and Len and all the research colleagues after the BMS acquisition because of the scientific foundations that drive the company that was sort of like Medarex on steroids. And basically recognizing at that time from the beginning that the key to really building a strong immuno-oncology portfolio lies in combinations. And we began to sort of build a whole effort in, wide, range of models and looking at various targets and concepts, and recognize first that an important component of any credible immuno-oncology portfolio would be a PD-1 inhibitor, not necessarily because it's going to be better than the other ones out there, but to provide a foundational element that can combine with other agents. And on top of that, we also sought to look for what would be appropriate combinations. So we looked at targets that fall into the regular sort of checkpoint inhibitor class, the TNF receptor agonist class. But we also, as Regeneron has always done, innovated with a new technology and started developing bispecifics in a way that I think really stand out from the rest of the field. And it stands out because the bispecific antibodies that have been created are very close and essentially indistinguishable from normal antibodies. And the reason that's so important is because ultimately when you bring these things into the clinic, you know, it's easy to sort of make all these Rube Goldberg type of combinations of this binding region and that binding region. But at the end of the day, you need to turn it into a drug that can be administered to patients, that behaves with appropriate PK, that has appropriate stability and ability to manufacture. So being able to build this bispecific program on the antibody framework meant right away that in the long term it would have many attractive features that we think will play out in the long term. We also developed, and by we I'm taking credit for, or I'm talking about the phenomenal research that has gone on for 30 years, building the VelociGene mouse that resulted in VelociMune, and now VelociBi, which is basically an altered version of the VelociMune mouse that makes making bispecific antibodies very facile. And there are two flavors of bispecifics that have been generated, one targeting CD3 and one targeting CD28, called the co-stim bispecific. But let me get back a little bit more to where we are with our PD-1 inhibitor, Libtayo. So from the beginning, we chose to target anti-PD-1 rather than anti-PD-L1. We put our candidates through a very rigorous testing program to feel confident that we were coming in with our best foot forward with a really potent and promising agent. And we were able to actually, despite sort of coming in a bit later than everybody else, we were able to identify two different indications that had been missed by everyone else in the non-melanoma skin cancer space. The first, cutaneous squamous cell cancer, advanced CSCC, in which we have our first approval, which we achieved in essentially record time, a little bit over 3 years from first patient dose in our first in-human study to getting an approval. And most recently, we announced very promising results in a second-line basal cell carcinoma study for patients with who have now progressed or are intolerant to the hedgehog inhibitors, and at that point really have no approved option. And so that's where we want to start, with competing in areas that haven't been sort of the open space. But we've also, gone in, waded into the big market of lung cancer, and have been very pleased recently to announce our ability to stop our pivotal study in patients in first-line lung cancer with PD-L1 positive tumors greater than 50% by the Independent Data Monitoring Committee, who basically, after only 50% of the planned events had occurred, said, look, you guys have won, go forward. So we have stopped, the randomization in that study. All patients are able to get Libtayo, and we've met now with the FDA for both of these programs, the BCC and the lung cancer, and we're working hard towards filing our applications for these this year. So the basic core of the approach has been, let's get our PD-1 approved. Let's but show that it's capable of performing as well as any other PD-1 out there. And I think everyone knows that particularly in this monotherapy setting in first-line lung cancer, the field, despite all the other antibodies out there, the field doesn't have many agents in there. We're only the second anti-PD-1 to announce a monotherapy positive study. And we're looking forward also to our chemo combination study for patients with any PD-L1 positivity. We basically should be finishing enrollment shortly, and are expecting perhaps some initial results in 2021.
So Izzy, I'll let you finish. So I wanted to just stop you right there and kind of focus on the lung cancer data that you just referenced. Specifically the strong top-line data you presented, I believe, back in April. Then you had a modified intent-to-treat update on the coattails of ASCO. Can you explain to us what the difference between these two hazard ratios, the first one being 0.676, the second being an impressive 0.566 in that frontline monotherapy lung setting, kind of what are the differences, and how does this shape what you want to do with Libtayo in non-small cell lung cancer?
Well, of course, we were very thrilled with the initial observation of the intent-to-treat analysis. But when we started the study, we were blocked from being able to use the PD-L1 assay in the fashion one needs to use it for a pivotal study under investigational use settings. And until we were able to sort that out, we had to use a somewhat less reliable testing system. And therefore, the FDA asked us from the outset that, well, look, we want you to check your data with the test on any patient that had the test done per, up to snuff. So when we did that, what we found was that of the original 710 that we analyzed for our intent to treat, there are about 560 that had either initially done the test, up to snuff, or upon retesting, we found that they actually truly were PD-L1 positive. So by definition, then some of the patients turned out to not test positive when we retested them. And so in that, what we call now the modified intent-to-treat analysis, which is the intended population that we wanted to study, namely the patients with PD-L1 greater than 50%, we did that analysis and found that it was an even stronger hazard ratio. And we've talked to the FDA about this. We're hopeful that this analysis will be part of the label. And what it just tells to us, even our initial intent-to-treat analysis was in line with the best data that are out there. And, of course, the modified intent-to-treat analysis is even stronger. So what this tells us in this study that, was quite large as well, that we have a good antibody, that we have something now that physicians, hopefully if we get this approved, they will have a reasonable choice. And what we've heard from all the physicians that we've spoken to, and when we started to share some of our information about the positive outcome, they're excited to have choice. So we think this is basically what it means. I hope I've answered your question.
No, that's very helpful. And along those lines, where do you believe Libtayo can play a role in treating patients with non-small cell lung cancer? You had mentioned that this is really only the second PD-1 to have, a positive monotherapy trial. How does that data set shape where physicians could potentially use this asset?
Well, if we get the approval as we hope, then patients that are coming in the first-line setting who have a PD-L1 test that shows that they're greater than 50%, and the physicians are willing and think that it's appropriate for these patients to start on a monotherapy agent and delay or avoid chemotherapy, we hope that Libtayo will be an option. And as you know, that's a very large market, a very large patient population, so we're looking forward to many physicians getting experience with Libtayo and seeing how well it performs. And as I also said, we have a study ongoing to test the combination in both non-squamous and in squamous non-small cell lung cancer in the first-line setting, regardless of PD-L1 positivity, in combination with chemotherapy versus chemotherapy. And so we hope if that study is positive also, and obviously if the monotherapy study was positive, we're cautiously optimistic that too should become positive, that we will have, for any patient coming in first-line lung cancer, they'll have a choice.
Excellent. And then just shifting gears slightly, one of the themes from your ASCO call was the importance of combinations. Can you, on a high level, talk about how Libtayo can be the backbone for many of these combinations? And, what indications do you think would be most suited to a combination approach? And I know that's a very broad question. We can talk about the bispecifics in a moment, but on a high level, can you kind of introduce the topic for us?
So, on a high level, any tumor in which you think there's any opportunity to consider a immunotherapy approach or bringing to bear, a T-cell mediated and maybe in the future other arms of the immune system is fair game. Because one of the things that happens is what we've learned is that the immune system plays a very important role in managing the emergence of potential tumors throughout life. And the fact that tumors become clinically apparent typically occur because the tumor has outfoxed the immune system. What we've learned is one key mechanism that they do this by is by using the checkpoint blockade system inappropriately, if you will, to basically turn off an immune system that probably protects us for many years from tumors actually ever emerging to be clinically apparent. On top of which, if there are tumors that don't have antigens, either neoantigens or molecules that are being expressed in the wrong place and the wrong context that the immune system is able to identify as something fishy going on here, then we can think of artificial ways, or quote-unquote artificial, using the bispecifics, to really bring T-cells to bear on these with other approaches, which we will talk more about the bispecifics. Now, in terms of combination agents, we view this very broadly. We've learned that chemotherapy actually can combine with checkpoint blockade. I think people at the very beginning were concerned whether that would work or not. It does seem to work. There are other, molecules in the checkpoint family or in the TNF receptor family. So we have a LAG-3 program underway in combination with PD-1. We announced that we'll be bringing our own GITR antibody into the clinic this year. The IND for that has been allowed, so we hope to start that soon. Obviously, things get a little slow with COVID, but hopefully that will move forward. And then we have the whole combinations with the bispecifics. But before we talk about the bispecifics, I'd also just add that one of the unanticipated, to me anyway, surprises and benefits of us developing such a strong antibody has been that many companies have come to us with, seeking to partner with us. And it's put us in a position to really look at companies that have complementary technologies that are not in our wheelhouse, but where we, look at companies to make sure they're like-minded scientifically and they have compelling platforms as well as lead products. And so we've partnered now with several different companies making vaccines, oncolytic viruses, techniques to deliver cytokines, and we are continuously looking and evaluating opportunities in that way as well. So that was sort of an unexpected, benefit that we had as another way of going forward. But our main, excitement lies around our bispecific combination platforms. And I can go into that if you'd like.
Perfect. Well, along those lines, can you kind of walk us through your bispecific platform? You've mentioned a lot about the co-stimulatory bispecifics. How do those bispecifics differ from, CD3 targeting bispecifics, and kind of what is the potential incremental benefit that you could see in these patients?
Sure. So when T cells are triggered and activated to learn how to respond to specific antigens and generate and expand a T cell response that proliferates, expands, and is activated to actually kill target infected cells, which is what we think it was all originally meant for, but also tumors. It requires several signals, and I think, many people think, it's part of sort of a balance so that, you don't get overactive immune responses in autoimmune disease. So it requires signal one, which is the T cell receptor seeing an abnormal antigen in the context of the HLA molecules. But that signal one by itself, if it is not sufficient to really activate a T cell, in fact, if you initially expose a T cell like that to signal one alone, it can actually turn the T cell off. And we think that's partly how we learn not to attack our own tissues. So there's signal two, and signal two is mediated by any one of a family of what are called co-stimulatory molecules, but the most important one on the T cell surface is CD28. And it binds with molecules of the B7 family that are typically presented on antigen-presenting cells, so dendritic cells, macrophages, even B cells. These are the cells that sort of eat up antigen, present it, and educate and teach T cells to expand and get fully activated. And then after that, there's the class of signal 3, which can be cytokines that are also produce IL-2, IL-12, IL-15, etc., that drive further the expansion and activation. So what we've learned with the CAR T cells in particular was that, if you have a T cell that doesn't know how to recognize a tumor and you give it something to substitute for its T cell receptor, the chimeric antibody receptor, the CAR, you can redirect T cells to kill tumors, and that's been successful. We made CD3 bispecifics, because we didn't think it's great. The CAR T cells are great, and we've partnered with some CAR T cells, but in many events, many settings, it probably doesn't need to be that complicated. So we've made antibodies that are off the shelf, able to bind to the T cell receptor, the CD3 component, and bring it to bear on a tumor that has a particular antigen, or CD20 by CD3, or BCMA by CD3 in myeloma, or MUC16 by CD3, and et cetera, and more to come. But what has been noticed in the field is that these CAR-Ts and bispecifics so far have been active and successful more in hematologic malignancies than in solid tumors. And one possible explanation, I'm sure there are others, is that the hematologic malignancies share their developmental history with the same types of cells, so B cells and myeloplasma cells. They share the same kind of lineage as authentic antigen-presenting cells, and they typically express some of these B7 molecules. So it's possible that the bispecifics or CAR-T cells that target the hematologic malignancies are also getting the co-stimulatory signal. In contrast, solid tumors, which are generally epithelial or mesenchymal, like sarcomas, or breast cancer, lung cancer, melanoma, they don't make B7 on their surface. And so the only way for the B7 signal is if there are dendritic cells or macrophages in the tumor, and the tumor figures out ways to keep them out or to not make them work too well. So what the co-stim approach is that we said, let's make a bispecific that binds to a tumor target and also binds to CD28, and therefore turns an epithelial cell into a essentially pseudo B7 positive cell that is like an antigen-presenting cell and can deliver signal too. And the cool thing about this is that the way we've worked on these targets is that they seem to not work that well. They're not that active by themselves. And in fact, if you deliver signal 2 alone in general, it doesn't do anything. There was in the past a very unfortunate experience with the Regeneron antibody to CD28 that actually ended up being broadly stimulating for a variety of reasons and almost killed a few normal volunteers. And so people shied away from this. But we recognize that CD28 is key, and we made bispecifics that only activate and only transduce the CD28 activation signal in the context of the tumors. So as a consequence, what we found is that these co-stim bispecifics synergize beautifully with either CD3 bispecifics, making solid tumors now accessible to these T cells that are redirecting, and they also synergize with anti-PD-1. So if you have a T cell that does recognize a tumor but has been inhibited by the PD-1 pathway, if you have PD-1 there and it's still not enough, we can augment that by using the co-stim bispecific. And so these are two pairwise combinations. There's also the CD3 and PD-1 pairwise combination. And in the future, we could, entertain all three if it's required. So this is the kind of plug-and-play combinatorial opportunities we have that we are really excited about.
So I want to focus a little bit on the MUC16 by CD3 bispecific in ovarian. And, can you comment about why MUC16 in ovarian, and how do you get that potential utility of a CD3 in solid tumors, given that we've seen it usually work better in liquid tumors?
So MUC16 is the cell surface molecule that, when it's clipped, gives rise to CA125 in the blood, which is a typical marker that is used to follow ovarian cancer. And so it's very broadly expressed on ovarian cancers. It's also expressed on a number of other tissues, as well as some other tumors like some pancreatic cancers. So we've directed our bispecific to target the region of MUC16 that's on the cell surface even after clipping off the CA125 and linking it to CD3. We're in dose escalation now of both monotherapy and in combination with PD-1. And we, so far so good. We're able to deliver this, and it's been well tolerated. And we hope, maybe in the coming year, in 2021, maybe we'll have some information to share. But on top of that, we've just received permission to go forward with a MUC16 co-stim as well. So MUC16 targeting CD28, bispecific with CD28. So this is an example where if the MUC16 by CD3 by itself as monotherapy is insufficient to really mount a meaningful clinical anti-tumor response, we will have the ability to further augment it, or whatever it's able to do. We'll be able, we think, to further augment it with either anti-PD-1, Libtayo, or our MUC16 co-stim. So this will be an example of the ability to bring this to bear in one solid tumor. We already have our first co-stim in the clinic. It's a PSMA by CD28 co-stim for prostate cancer, and that study is underway in combination with Libtayo, and that dose escalation is proceeding. And we hope also next year to have some initial clinical data from that. And we should be, advancing more co-stim this year in both actually solid as hopefully liquid tumors as well.
And along those lines, your two kind of assets most furthest along are REGN 1979 and REGN 5458. Can you, for 5458, this is the BCMA targeting bispecific. That's a pretty crowded space. How does this asset differ from the whole host of other, drugs being developed targeting BCMA?
So I think the BCMA field right now, there are BCMA CAR-Ts, there are BCMA CD3s, and BCMA ADCs. We've been very pleased with the data that we've had to date with our bispecific CD3 by BCMA. We've seen right from our first cohort, some of the first patients we've dosed, a meaningful response in a patient who, has all the patients in our studies have failed all three classes of agents, IMiDs, proteasome inhibitors, and an anti-CD38, typically multiple different combinations of those. And, one of our first responders that we described at ASH last year actually had extramedullary disease, which is an even tougher setting, and this patient has actually remained on treatment and is having a great response. So it's, we recognize it's competitive. It's good for patients to have options as usual, but we think we have a very strong agent. And I think what we'll see as we go forward is that we will be looking to also explore combinations. So if, again, I think our BCMA by CD3 will be competitive. We are rapidly expanding our dose escalation, and hopefully we'll be initiating additional trials that will have registration intent. So stay tuned for that. That's all I can say on that for now.
And before I want to have Justin just comment on the opportunity back for Libtayo and basal cell carcinoma, one of the indications that you're pursuing. Any final comments on the oncology program? I know we could talk for hours and hours and hours because of the variety of combinations you have, but anything that you think people need to fully understand better, or you want them to, internalize when it comes to your work in oncology?
So,
sure, happy to, Evan.
Basal cell in particular or in general?
Well, I'm just talking about in general on kind of what we've been talking about. Then I want to pass it over to Justin just to comment on kind of some of the commercial opportunities.
Perfect. So we basically have come forward with, trying to bring forward a deck of cards, basically, where we can have multiple winning hands, and that we can, we know the agents. We vet them internally. We have great technology underlying them. All of the, antibodies are simply not commodities. I mean, people sometimes think of them, it's easy to make an antibody. Well, yeah, it's easy to make an antibody, but it's pretty hard to make a very good antibody. And we make very good antibodies. And we have really, I think, been able to now develop a tripod, at least, of foundational technologies, checkpoints, CD3 bispecifics, CD28 co-stim bispecifics, and there are more in development looking for other ways to engage the immune system to bring it to bear on tumors. And so we think we have this combinatorial flexibility that as we go forward, we think we will have, really winning combinations to bring forward to treat cancers.
Excellent. And then, Justin, so from the commercial perspective, I know you're making a lot of progress with Libtayo. Can you talk about what you think about the market opportunity for Libtayo and basal cell carcinoma following the data you top-lined, a couple weeks ago? And then also talk about where you think Libtayo can compete in the lung setting.
Sure, happy to, Evan, and I'll try to do so quickly given that we have Neil and Bob on the call as well.
Exactly.
I think the first thing to add is just that basal cell carcinoma is on the order of a few thousand patients in that second-line setting for patients who have failed a hedgehog inhibitor. And the data is really remarkable in that not only do you see response rates in 20 to 30% range, but just the high proportion, 85% or so, who are maintaining a response after 12 months is just, it's staggering as you think about just the opportunity that patients can have post a hedgehog inhibitor. But I think if I were to leave you with just a couple of thoughts here, more broadly, commercially speaking, Evan, I think one is that, as you heard from Izzy, we're very much focused on the long term here. We have a tremendous pipeline that is going to offer us flexibility to get after cancer in ways that a lot of other companies have not been able to date, and really believe that our pipeline positions us very uniquely in that regard to help more patients, both in cancers that respond to PD-1 blockade, but also in patients and cancers that don't necessarily respond. So that's very much the long view here, and as you heard from Izzy, we're beginning to reap some of those benefits. But, I think there's always the focus on the short term, and to just round out your question on how do we think Libtayo can compete in the short term, I think it's just important to make sure everyone's clear that physicians, by and large, are fairly heterogeneous. They make decisions that are appropriate for their specific patients and the type of disease that they see, or how they believe certain drugs will work in certain patients. And so there's definitely a personalization aspect to treatment that I don't think can be understated. And so when you can bring a new drug into a large market like lung cancer and offer physicians a choice for a monotherapy that they really have not had to date, that is going to be meaningful, particularly when you can bring along with it a very strong overall survival benefit. And so while we do have a long-term view, we do believe that we can be competitive on the merits of the strength of the data and what we see in terms of being a very strong drug within lung cancer and some of the other skin cancers that we've shown to date.
Excellent. With that, I want to turn the program to Neil. Neil, are you there? Right.
Yeah, I'm here.
Neil,
Hi.
Perfect. Hey, how are you? So I don't believe people have met you or have been introduced to you much on the phone. Can you give us a little bit of your background and kind of talk about what you're doing right now with the COVID-19 program? Then I want to just jump right into questions.
Sure. I've been at Regeneron for, this is my 29th year there, so I started when it was quite small. And I started in the discovery research area and did a lot of work on the mechanism of cytokine receptor signaling. And I actually was the inventor of our trap technology, which turned out to be the basis of our first 2 approved drugs before we developed the capability to make fully human monoclonal antibodies. I also built the entire preclinical group that has responsibility for making the antibodies and making cell lines out of the antibodies, doing the process development that's required to scale them up and give them to our industrial operations group, our IOPS group, the PK group, the formulations group, the toxicology group. And I have to say that I think that we put together the best operation end-to-end of anybody in the business. And, at Regeneron, we've always focused on, as Justin mentioned, the long term, but also on 2 things: very strong and deep basic research coupled with technology development that allows us to innovate around bottlenecks in the whole process of discovering a new drug and getting it rapidly to patients. And I think the perfect example of that is our efforts on these emerging infectious diseases. So we made fully human antibodies with our VelocImmune platform, which is our mouse that was engineered using VelociGene, and it's the largest genetic engineering project in the history of the world where we humanized 6 million bases of the mouse genome to allow them to make fully human antibodies. And they have a wild-type immune response, and it's our go-to platform that we've made every single fully human antibody that we put into the clinic up till now. So for Ebola, we did the entire project of starting the project and isolating thousands of antibodies that bound to the external protein, the glycoprotein of the Ebola virus. And we ended up picking 3 very strong antibodies with complementary mechanisms that bound independently to the virus. So they could all 3 bind at the same time, which, as I'll get to, is a really important feature. And so we did that whole project in 6 months. And then when the coronavirus outbreak happened, the viral sequence of the genome was actually published on January the 12th, and we started the project that day. And within 5 months, we had screened through thousands of fully human antibodies and isolated hundreds of very strong neutralizers, both from our VI mice as well as from convalescent humans. So we had antibodies that we got out of people that had COVID, and so we were able to look at both modalities and pick the best antibodies from each to combine together as a cocktail, so to speak. And so the spike protein for coronavirus is a little smaller than the glycoprotein for Ebola, and we ended up with 2 complementary antibodies that bind independently to separate places on the virus. They both independently neutralize, and together they will hopefully prevent virus escape, which I can talk more about.
Perfect. So where is Regeneron currently with the development of the antibody program? I know that there was a recent announcement of the initiation of clinical trials, but where are we and when could we see initial clinical data from this program?
Right. So we announced that we started trials a couple of weeks ago now, and that is in the setting of patients that already have COVID that are either hospitalized because of their severe symptomology, or they're ambulatory and their symptomology, although they're positive, is not bad enough to make them be enrolled in the emergency room. And so we have both of those settings right now. And at least for the patients that are in that setting, I think we're going to start getting data that's meaningful in the next few weeks, and we should have data, I think, this summer for sure, because there is no shortage of patients in the hospital still, or with COVID positive that are ambulatory. telling us that the antibodies are safe with some initial readout on effectiveness. And we're looking both at the ability to inhibit viral shedding from positive people, as well as to resolve symptomology in people.
And you had mentioned the importance of studying viral escape and kind of how the antibodies prevent that. Can you expand on that and talk about why that's so important and what it means for this program and how we actually get out of this pandemic?
Okay, so you know that for HIV, the experience with HIV is very clear-cut, that the initial efforts against HIV were with single drugs, and the virus, which is an RNA virus, rapidly mutated and was able to find mutations that would inactivate the mechanism of the drug, but still allow the virus to be functional. And so it turned out that a combination for HIV of 3 independent drugs was the best way to suppress the ability of the virus to generate escape mutants. And the same thing is true of the coronavirus, we believe. And I believe so far we're the only people that have done real experiments using a pseudotyped virus. So what it is, it's a VSV, vesicular stomatitis virus that you can use in the lab, that we put in the spike protein so that it infects cells just like a coronavirus would. And then we're able to do very rapid experiments then to see if with a single antibody that virus can generate mutations in the spike protein that will allow it to still be functional but escape the neutralization of the antibody. And we published in Science papers a week ago today, actually, in the journal Science, that with a single antibody we were able to find escape mutants, but that with a combination of 2 antibodies that we did not find any escape mutants. And moreover, we were also made separate viruses with individual mutations from the escape mutants and showed that our cocktail of 2 antibodies would actually also neutralize those. And so I really think it's a really hugely important thing, because we don't want to have treatments generate escape viruses out there that become inactive for the treatment you've given them to. And so I would just implore other organizations that are looking at single antibodies to do these escape experiments and make sure their antibody is not subject to escape. But so far, we have not found any antibodies that are able to suppress escape mutations on their own.
Excellent. I want to just pause right there, and I'll come back in one moment. Operator, can you see if anyone on the line has any questions? Can you read the instructions?
Ladies and gentlemen, as a reminder, to ask a question on the phone line, you will need to press the star and the 1 key on your touchtone telephone. To withdraw your question, please press the pound key. Again, to ask a question, please press the star and the 1 key.
And while we're putting together the question queue, Neil, can you talk about the difference between your primary cocktail and the backup antibody cocktail? Kind of some of the differences between the two of them.
Because we had so many antibodies available to us, we were able to make multiple cocktails. And we've actually have looked at structures of where they bind on the viral spike protein. And so we picked the ones to start with that we thought were a very strong cocktail with strong neutralization. But should that one show any escape or stumble in any way, or we're not happy with the efficacy, we have a backup cocktail and with more antibodies coming behind those that we could switch to that have different binding sites and would, I think, have independent properties.
And when you look towards the clinical trials, what's the bar for efficacy that you want to see? How quickly do you want to see symptoms resolved? Do you want to see a mortality benefit? And how do we think about what type of benefit you can measure in the prophylaxis setting?
Well, for Ebola, what we saw was that, and some of you may recall that last year in our Ebola efforts that we were involved in a study in the Democratic Republic of the Congo on patients with Ebola that was a randomized controlled trial under incredibly challenging conditions of these people out in a remote part of Africa where there was actually some local violence going on. And so they deserve a huge amount of effort for getting that done at all. But in that setting, they stopped the trial early because our drug provided it hit a pre-specified boundary for superiority over another triple antibody cocktail, which was ZMapp. And in that study, the amazing thing was to us was that in patients that were treated within 5 days of developing symptoms, there was a 90% survival rate compared to about a 30 to 35% survival rate in untreated people. So at least in that case, we were able to get a really resounding increase in survival. So in this case, we'll have to see what happens, and it may depend on how early we get treatment. We certainly believe that the earlier you get treatment, the better, and so it may turn out to be a mix depending on how progressed the patients are when they actually get treatment.
Excellent.
I should also add, I can add one more thing, is that we did have also an antibody cocktail against MERS, which is a related coronavirus to the current COVID-2. And in animal studies, we were able to totally cure mice that were already infected with MERS and totally prevent disease in mice that were given the drug before they were infected with the virus. So at least that's one data point we have with another coronavirus.
And operator, I believe we have one question in the queue.
Yes, I'm sure we have one question coming from the line of Diana Belsky with Viking Global. Your line is open.
Hi, thanks so much for the talk. I was wondering if you could comment on the dosing that might be required for hospitalized patients versus early patients versus prophylactic, and if you have any updates on guidance for how many doses you can provide in the fall and beyond. Thank you.
We haven't really disclosed hard details about the dosing, but certainly we are using 2 different dose regimens in both the treatment and the prophylaxis setting, and we're using a higher dose in treatment than we are for prophylaxis. You may know that the virus, once there's virus on board, will actually bind the antibodies and accelerate the clearance of the antibodies. So in general, you have to give more for treatment, but we haven't talked about the specific doses yet. We should be able to crank out as many as a couple 100,000 doses a month right now for the prophylaxis type of setting. And we're trying to shift some of our manufacturing to our facility in REGN in Ireland so we can make more doses here in the States. And we're also looking to see if there's more capacity out there that could make more drug in anticipation that it's actually going to provide some benefit.
Excellent. And I have one more question on the COVID-19 program, then I want to just turn the call over to Bob for the final few minutes of our conversation. This has been very helpful for all of you on the call. Can you discuss the rationale for the current adaptive clinical trial design for the program? I know that was something you highlighted in the press release. What does that mean, and how does that potentially speed up the development effort?
Well, I think the first part of the adaptive design is, obviously these antibodies have not been in people before, and so we have to get some basic safety information. I think we're all very optimistic that the antibodies will be safe because our experiences and the experience of the field in general is that if antibodies are aimed at non-endogenous targets, like this is a external viral target and it doesn't bind to anything in a human, that they are very safe unless for some very rare reason there might be some infusion reaction or something like that. And so we're optimistic on safety, but we have to establish safety and get some initial data, I think, on frequent viral clearance measurements and give us maybe a better idea of the doses we need to go into a second phase 2. And so that's the general idea of the adaptive study.
Excellent. Any final comments on the COVID program before we shift gears to Bob?
No, I think that's the top important line, so let's go to Bob.
Thank you. Bob, I would never forget about you. Are you on the line?
I am. I'm here, Evan. Ready to go.
Great. I'm ready to go. Perfect. Well, you've, as Israel and Neil, have had a very busy first half of the year. So have you. You've restructured the accounting presentation for this, all the collaborations. You've executed a share buyback. So can you just kind of walk me through, now that you've done this share buyback, the Sanofi secondary has already occurred, what's your capacity for business development, and how are you thinking about capital allocation going forward?
Yeah, thanks, Evan. And I think, Izzy gave me a good setup for this when he began, the conversation with regards to, coming over from MetaRex, and then obviously everything that Neil built, all the pre-clin stuff that Neil talked about. I mean, there's we see no better investment than within ourselves right now. And people look at, percentage of revenues, and they look at our R&D number, and, sure, it's higher than most, but that's for good reason. That's because we make the conscious decision that, we really like the technology that we have, and we like investing in ourselves. We think there's no better return on invested capital. So, despite what people saw in the last couple of months with regards to the buybacks and all that stuff, first and foremost is within R&D, within the Regeneron shop is where we are going to apply our capital first and foremost. And then secondly, we are seeing good opportunities with regards to BD, but, we look at BD kind of not at the M&A initials, but more as kind of tuck-in technologies. I think Izzy or Neil said like-minded science people. I mean, we try to find companies that can be additive to all the platforms that Neil built, right? What do we not have that, 1 + 1 will give us 3 with regards to our antibody technology? And, we haven't been afraid to do stuff. Recently, we're coming off of the heels of doing an Intellia transaction. Izzy talked about Viriad on the oncolytic viruses. Certainly, we're really pleased with the Alnylam transaction that we did in the technology, their kind of intracellular technology that we didn't have access to, that we will now have access to, and we're going to be bringing a C5 program into the clinic very, very shortly on that end that we're very positive on. So, BD will be second. We're not out there trying to get in a Tesla for accretive cash flow or, fill a phase 3 because we're missing this or missing that, or a phase 2, really kind of early tuck-in technologies is really what our focus is. And then, lastly is return to shareholders. When we see opportunities like we've seen, particularly back in November, and then even this $5 billion buyback with regards to, tacking it onto the secondary offering and knowing, that the price that we're going to do this at was going to be at a, 510 or 515 or 505 handle. I mean, we liked that. We liked that valuation, in terms of the intrinsic work we've done with regards to where the public is seeing us. So, we kind of put our money where our mouth is, went out with the $5 billion buyback. And on top of it, we were able to kind of use a little leverage, which is a word that, Regeneron hasn't used before with regards to its balance sheet. Take advantage of the capital markets that are out there. Right now, we have a fully financed bridge loan that we will replace either with a bank club loan or, we'll do 5s and 10s. And, we're working hard to kind of get to the rating agencies and take advantage of good market conditions.
Right. I was pleasantly surprised when I did hear the word leverage come out of your mouth with regard to the loan. So looking, can you comment as to kind of the strategic rationale behind the $5 billion share repurchase? That also seems to be a deviation from the Regeneron capital allocation strategy from, years ago.
It did, Evan. And I think, kind of first and foremost, you need to have conviction in business fundamentals. We had nice kind of tailwinds coming off of, unfortunately, the Bayview situation. What Izzy's been able to accomplish on non-small cell lung, stopping that trial early, BCC. And we really like the future prospects for our commercial and pipeline products, and therefore we like the valuation. Certainly it provides immediate accretion. Rarely does a CFO have an opportunity to kind of take 7%, shares outstanding off the table in a nice, smooth transaction. So that was great. And again, it did allow for a kind of easier placement of shares by us being part of it. We were able to get these shares into really sticky hands, so we're really kind of pleased with that transaction. And I think, Evan, the way to look at it is, it was kind of I hate using the word one time because one time becomes old when you do it the second time, but kind of a one-time opportunistic trade, that we had good tailwinds to do it. The capital flows were good in the biotech business, right? IPOs were going well. We knew the secondary offering was going to get placed. We had this damn overhang that we needed to get rid of. Paul Hudson, the CEO of Sanofi, was very clear that this event was going to come upon our existing shareholders, and we just didn't want this overhang out there. So kind of a culmination of all those facts led us to our decision to do that.
Right. And then talking about the L word, leverage, how do you expect to continue using leverage to invest in the business, form these early stage partnerships? Is that going to become part of your capital, your balance sheet management strategy going forward?
Sure. I mean, certainly my background as treasurer of Pfizer and Wyeth, I mean, leverage is your friend if used properly and in moderation, so I'm pleased that we're going down that path. And again, we'll be good stewards of our cash and marketable securities, of our shareholders' spend going forward. So, kind of rest assured on that. And, the other use of the word leverage is certainly getting good, operating margins and the like. And, we do expect with Dupixent going forward, all the indications, all the geographic locations, all the different age groups, that as more and more gets put out there, again, as you know, we had an important end of May PDUFA filing that our FDA acceptance with regards to AD for the younger kids. We expect to get, better and better leverage as it associates to our Dupixent and our Alliance brand P&L. So kind of two good uses of leverage, one for the balance sheet that, we'll have to do it in moderation, and then certainly one for the P&L in terms of just getting better leverage, as more and more sales occur and we have the infrastructure set up from an SG&A perspective.
And we are about 2 minutes past the hour, but I have two final kind of questions for you. One, have you started to see kind of the benefit of the collaboration restructuring in terms of investors understanding the business better? And do you think that there is still things that you can do? And two, 6 months from now, with kind of the business going as it is there anything else that you'd like to change or kind of evolve in terms of the way you manage the P&L, the balance sheet, or kind of the way you look at capital allocation?
I think on the first question, when we redid our accounting and kind of recast the numbers, we were just trying to ensure that there was transparency, right? People that go deep into Regeneron, they know our operation, but there's a lot of people that don't, in today's day and age, all biotech companies don't have the time and energy to be able to go really deep. So I'm happy that we changed our accounting and provided, what we think is a really transparent scene going forward. But with that transparency, Evan, comes exactly to the question you're asking. What do we need to do now? We need to execute, because we're laying out within our MD&A section of the 10Q or the 10K, we're telling you what our kind of margins are. You can see them within the Alliance. And I'll tell you, in Q1, we did still have Kevzara and Praluent. We had losses in both. So, again, back to the leverage word, we hope to get better leverage on our margins going forward. 6 months from now, it's the continuation of delivering, right? Ensuring that what we put into the clinic, are fully tested and viable antibodies that are coming from Neil's shop. I mean, it shouldn't be a change of pace from anything that we're doing. We feel, we feel good in the position that we're currently in, and need to continue to execute on all fronts. All right, so why don't we stop there, Evan? I think we're over our time.
Sorry, I was on mute. I just wanted to say thank you to everyone on the call. I really appreciate it, Bob, Izzy, and Neil, and Justin for the time today. And if you need anything, I'm in the virtual office today. Have a great one, everyone. Bye now.
Thanks, Evan.
Bye-bye.
Ladies and gentlemen, that does conclude our conference for today. Thank you for your participation. You may all disconnect.